Dr. Messersmith, you can take it away.
It's a pleasure to be here. So I would like to spend a few minutes talking about my work on muscle-inspired adhesives. This is actually something I've been working on for over 20 years now, which dates me in my age. I thought it'd be a good idea to start with a few simple definitions, and these, I will admit, are directly from Wikipedia. Okay, so biomimetic and bioinspired, what do they mean? So biomimetic means the concept is to emulate models or systems or elements of nature for the purpose of solving complex human problems. Whereas bioinspired is a subtly different, but my interests are more aligned with bioinspired. And the idea here is that you're inspired by nature, but not necessarily limited by what you observe or see or understand from nature. And a good way of explaining that would be, in nature, organisms solve problems in very restricted and confined and prescribed environments. Whereas what we can learn about how they go about that could be applied to many other situations, okay? And that's more in terms of inspiration, that's more what we do in my group. So we have in mind to learn from biology, apply it in a variety of different ways, which, in nature never intended or thought about. My own particular interest has been in muscle-inspired materials, and so these are specifically marine mussels, those that you find in the sea. There are, of course, freshwater versions of these, but many people know mussels as being basically, something you might get in a restaurant. But if you've ever been to the seashore, you may have seen, particularly in North America and other places in the world, you may have seen mussels attached to rocks. And this is the part that has fascinated me for, as I said, over 20 years. They actually attach to each other. So the first layer of mussels attaches to something like a rock, or it could be a pier or ship hull or any structure in the marine environment. And then other layers attached to the mussels. So you have these clusters, and it's kind of hard to see how they do this, but the picture on the upper right of this slide shows this a little bit better, which is a mussel attached to a shiny surface here. And the interest of my bioinspired work is this structure, which is called the mussel byssus. It's just basically a collection of fibers, each of which is terminated by a pad of glue, if you will, where the glue attaches to whatever that surface is. And the fibers all come in and converge just inside the shell to the soft tissue of the mussel, and it keeps it. There's a muscle that keeps all of these fibers here in tension. And so this is the entire attachment structure of the mussel. This is how it survives in a turbulent environment. And back to the idea of inspiration, which is that we feel like there's a lot to learn about how to accomplish adhesion to wet surfaces for a variety of potential applications from learning about the mussels. And so we have been studying this for quite some time. This is an electron micrograph. Lower right is an electron micrograph picture of one of these glue pads, and you can see the fiber that comes off of the glue pad, and then eventually ends up going into the inside of the mussel shell. This is where the action is as it relates to adhesion. So these are, I'll tell you a little bit more in a minute, but these are very specialized glue proteins that the mussels secrete. But first, I want to show you a video or two, actually 2 videos of this process. So what I would like to show you is a video of one of these being formed in real time. It takes about 2 to 4 minutes total to make one of these attachments. And so this video that I'll show you here is basically going to show one of those. The video is going to show secretion of one of these fibers and the glue pad from start to finish. So we have an aquarium in the lab, and put put mussels in there, and we can take a video through a piece of glass. So the mussel here is sitting inside and immersed in water, and it's going to attach to a plate of glass that you see. So we're taking the picture through a plate of glass. This is what is called the mussel foot. This part of the organism that is responsible for secreting these fibers. So the first thing it does is actually comes out of the mussel shell and basically probes around on the surface. When it finds a place it wants to secrete an attachment, it will press hard on the surface, and that's what you're seeing right here. And so the mussel foot has glands that store the protein, and then in liquid form, and then it injects the protein into a little cavity directly onto the surface, which makes this glue pad, and then it also injects protein to make the fiber. There's a little groove that runs along the length of the foot pad, and then those liquid proteins actually react and solidify into solids in about 1 or 2 minutes. And then at the end of that process, after it's solidified, the mussel foot will pull away, and you'll be able to see this newly formed adhesive pad. You see, this is the glue pad on the surface, and then the fiber that goes back into the mussel shell. So that, for me, is super fascinating as a material scientist, and when somebody who loves nature. This is very elegant processing of a material. There's no other purpose for this tissue in the organism except to anchor it onto the underlying surface. Now the mussel, as you can guess from some of these pictures I've already shown you, does this many times. And the second video, this is much shorter. I want to show you the video of a capture of a mussel secreting 4 or 5 of these threads over a 4 or 5 hour period. So one thing I want to point out before I show you that is there's a very clear rubbery material. It's a silicone material that is on the surface, and between the surface and the mussel. The rubber bands that you see here going back and forth, these are actually just to help attach the mussel onto, or put the mussel onto the surface. And actually this experiment, we were trying to understand if the mussel would prefer the glass surface, which is out here, or would prefer to attach to this soft rubbery material. So let me start the video now. And so this is a frame grabs every 30 seconds over a many hour period. So you see the mussel foot come out, and one curious thing is that it pokes around over here and then finds that it doesn't like that surface. And then it ends up secreting these attachments over here. You see a couple of these threads that have been secreted, and eventually it falls off of the rubber band here. So it made, in about 4 hours, it made 4 or 5 of these threads. And then the mussel does that many times, and it depends on the season, the weather conditions, etc., how many of those threads it makes. So it's really fascinating materials processing of a glue that works well underwater. And to preface the rest of my talk, which is that's no small accomplishment. Many man-made adhesives don't perform well underwater. Okay, and so this is something that we believe that we can learn from. So I'll tell you a little bit about the biochemistry here. It's very, very complicated, but I'll try to make it very simple, which is these proteins, particularly the glue proteins, and this is a cartoon, if you will, of one of these glue pads. It has about 6 or 7 proteins that are known in the structure, and they are distinctive for having an unusual amino acid. So that amino acid is called DOPA. It's derived from another amino acid in the proteins. Some of these proteins have very high concentrations of DOPA. So the red Ys here that are underlined are all DOPA amino acids. And it's a remarkable feature of these proteins that they have so much DOPA. Elsewhere in mammals, including humans, DOPA is not found in proteins. It's only found in the marine environment and used in organisms that really have to attach to wet surfaces. So it has very special properties, and it's a very versatile glue, which gives these proteins their ability to attach to surfaces. And so we have, over the years, learned a lot about DOPA and its contribution to adhesion in these proteins. And then the rest of my talk, I would like to tell you about how we use that information. And one of the things that we've learned over the years, if you look in the upper right corner of this slide, which is the element of the dopa amino acid, which is special as it relates to adhesion, is this thing right here, which is called a catechol. Okay, it's a aromatic benzene ring with 2 hydroxyl groups. That's a very, very special and unusual chemical feature of an amino acid, and its presence in these proteins allows the muscle to secrete and attach to surfaces very effectively. So my group, what we do, and this is, you know, embraced in the idea of bioinspiration, we, at the end of the day, we synthesize synthetic materials that are inspired by these proteins. So we don't actually make or use the proteins, but we make synthetic polymers into which we chemically synthesize and install that catechol functional group that we have found in muscle adhesive proteins to be really important. So we can make a variety of architectures and compositions of molecules, and then hopefully at the end of the day, we can use them in several ways. One is to actually study muscle adhesion. So I won't talk about that this afternoon, but we can use these as model systems for understanding muscle adhesion. And then we can also think about translating these polymers in the form of medical or consumer adhesives, and I'll give you a couple of examples of those. In the area of basic research, we were approached by a group from Caltech to, they are studying jellyfish locomotion. Of all things, and they wanted to attach electrodes onto the jellyfish. So these are the electrodes that you see here, and they needed a water-resistant glue. So they came to us, and we sent them some of our synthetic muscle-inspired glue, which is being used here to attach these electrodes onto the cap of the jellyfish, and they're using that to study, you know, ask basic fundamental mechanical questions about locomotion of jellyfish. So that's an example of how our work can be applied to basic studies. I want to tell you about 2 medical applications just very briefly. One is upper left here, which is a potential treatment for diabetes, which is the idea of transplanting islets, which are the clusters of cells in the pancreas which fail during diabetes, and which can be implanted into humans as a treatment for diabetes. So that's one application, and then I'll talk about also fetal surgery. Actually, I'll do fetal surgery first. Okay, so fetal surgery. What is fetal surgery? Fetal surgery is a growing surgical discipline which it involves surgical interventions or surgeries done on the fetus in the womb, usually in the 2nd trimester. There's a couple examples of treatments that are done for various conditions. Spina bifida can be treated by operating on the fetus in the womb in the 2nd trimester. Also something called TTTS, which is twin transfusion syndrome, which is a condition where you have twins in the womb. And there's an unequal supply of blood to the 2 twins, and this is one of the surgeries that is done also to rectify the uneven blood flow. And both of these approaches to treatment involve or require access into the womb. And for this, you have to basically break through the fetal membranes, this is what we call the amniotic sac. Okay, and this is often requires something on the order of 3 or 5 millimeters. So it's a pretty large defect in the womb. This has been practiced since the early 1980s. This guy right here, Dr. Michael Harrison from UC San Francisco, he was the first surgeon to ever do one of these procedures on a fetus. In fact, this is a picture from 2005 of Mike examining that baby that was the first ever fetus to be treated by him and successfully brought to full term. And that patient was at that time 20-something years old. So it's an incredible story. And now these procedures are practiced as very specialty procedure. And the reason it's limited in its extent is because there's a lot of risks to this. And one of these risks is involving the penetration of the amniotic sac. And the reason for that is the amniotic sac has almost no healing capacity. So once you poke a hole in it, it does not heal. And this has significant risks for the fetus, okay? You have the possibility of leakage of the amniotic fluid out, bacteria getting in, and also a traumatic rupture of the membranes. Okay, and this has been referred to as the Achilles heel of this surgery procedure, because it's one of the things that really holds surgeons back. And to get an idea of why this is the case, if you look at a plot of percent survival of a fetus versus gestation. So full gestation is 39 weeks out here. Could say that's a full 9 months. The surgeries are done usually between week 16 and 26, so in this gray shaded area. And for a normal fetus, the percent survival, if they're born prematurely during this period of time, is very low. Okay, and so if this procedure induces some harm and results in premature birth, the survival rate is very low. Okay, and so there's a great need actually for adhesives to seal these during the surgery procedure itself. And that's one of the things that we have been doing now for about 10 years. Here is one of our synthetic polymers. I'll just point out the functional group that is found, the catechol that is found in the muscle adhesive proteins. We've now put this into a synthetic polymer and made a glue out of it. This is a collage of pictures taken of a human amniotic sac that has been repaired with one of these adhesives. Okay, so the purple material here is our glue, muscle-inspired glue. Here's the defect where the puncture was made, and that's several millimeters. And the glue here attaches well to the wet tissue and bridges this defect, and was good enough to withstand 120 millimeters of mercury, which is more than sufficient for third trimester intrauterine pressure. We also conducted a in vivo study in rabbits, where we showed that the survival rate of rabbit fetuses was increased from 36% if you just do the puncture and do nothing. 36% of the fetuses survive, and we could increase that to 80% with our adhesive plus basically a little plug that we glued in with our adhesive. So from 36 to 80%, very promising. We're still working on this, and currently I can just tell you that one of our approaches is to actually put in the adhesive before the surgeon actually breaks through the membrane. And this we call pre-sealing. So this is work that's ongoing. These surgeons are remarkable at using their surgical tools. So a surgeon can actually get into this little space between the amniotic sac and the uterine wall and put our adhesive in there, and then allow it to solidify and provide mechanical support for the actual intervention. So we're hoping that this turns out to be a even better approach. And then I'll just wrap up by saying that we're also interested in some things related to consumer adhesion. And the one that we've done the most work on is illustrated here, which is the very common type of adhesive, which is called pressure-sensitive adhesives. So these are pretty much any kind of tape or membrane that is glued down onto a surface. So Post-it is an example of adhesive, Scotch tape, etc. These are all pressure-sensitive adhesives. So basically, the idea here is you have a backing, some sort of a backing material. In the case of the Post-it, it's paper, and then you have an adhesive layer, and the pressure part of PSA is that it's just basically you press with your finger. Usually that's sufficient to adhere to the tissue, to the surface that you're trying to adhere to. These are often, not always, but often are synthetic polymers, and we're interested in making these better performing underwater. Most of these actually are not great underwater, and I can illustrate to you one of the polymers that we have recently made. Sorry, there's a lot of data on this slide, but basically here's the polymer. It's a pressure-sensitive adhesive polymer with, again, the muscle inspiration is right here with the cuticle in that polymer. And then we measured the strength of adhesion of a little particle of either glass or polystyrene or polyethylene to this polymer surface, with and without the muscle inspiration. And you can see if you just add a few % of that muscle inspiration underwater, you can increase the adhesion to glass several-fold. It works a little better for polystyrene, and we still have a lot of work to do for polyethylene. So it really doesn't have much of an effect on polyethylene adhesion, but it does significantly increase the adhesion, wet adhesion strength to oxide surfaces. And I'll just show a cute little video that we made of this. You may have seen this in the stores. There's something sold by 3M, which is called Extreme Post-its. They're advertised as water-resistant sticky notes. Let me restart this, go back to the start. Here in the video, this is a Post-it Extreme note we bought from the store, put a little weight on there, and it falls off underwater. And we made our own sort of variation of this with our muscle-inspired polymer. And you can see here a 500 gram weight underwater. Our muscle-inspired sticky note performs much better. So I will wrap up. I think I've used most of my time. So just to summarize, in general, we're working on bioinspiration, taking ideas and concepts and models from nature and applying them to synthetic materials for a variety of applications. I told you about medical adhesion and consumer adhesion, and we're continuing to work on this, and hopefully some of these things will end up either in your grocery store or as healthcare materials for the future. Final thing to say is that I appreciate the hard work of my research group. This is our annual party at the beach where we pull mussels off the rock and cook them up. And then I just wanted to say, because it's important these days, that taxpayer money is really important. Almost everything that I talked about was funded by National Institutes of Health. So your taxpayer money is hard at work and making a difference for us in academic research. So with that, I'll wrap up and thank you for your attention.
Awesome. Thank you so much.